All right, well, we will go ahead and get started. Welcome everyone to this research update on gyrate atrophy. I'm Karobi Acharya. I'm here with my husband, hello, Malay Acharya. We are the co-founders of Conquering Gyrate Atrophy, which began about 2 years ago. And we are so excited to bring to you this first ever that we have put together a research update. And the way that it's going to work is I have a few questions and then we will have time for audience Q&A. I am incredibly grateful and honored that we have Jean Bennett and Katherine Uyhazi joining us today and to tell us a little bit about the research that they are doing. And I want to just make it clear that the reason that that we're able to support this is because of all of the people who have donated and supported this effort in a variety of ways, both financially and otherwise. And so incredibly thankful to all of you who have helped with the fundraising and supported this effort. Without further ado, I think we'll go ahead and get started. And I'm not going to read both of their very extensive bios, but that is on our website, and you can check that out. But Dr. Jean Bennett is a professor of ophthalmology at the University of Pennsylvania, and Dr. Katherine Uyhazi is an assistant professor of ophthalmology also at the University of Pennsylvania. And a huge welcome to you both, and thank you for taking time to speaking with this community today.
It's both of our pleasures, and it's really amazing to see so many people online, and thank you, Karabi and Malay, for arranging this. We are so grateful to you, and it's great to be part of this big team.
Yeah, thank you so much. We're really delighted to be here today. It's great to see so many of you joining us, and we're really grateful for the support from Conquering Gyrate Atrophy, which has really allowed this research to progress.
Wonderful. Well, thank you. So, like I said, I'm gonna start with some of my own questions that I think will be of interest to everyone. But there will be time for audience questions. So, if you have questions, you can enter them into the chat. And there may also be an opportunity to even ask them live, depending on our time. So, all right, well, so let's get started. Jean, this first question is for you. Many of us on this Zoom have been affected by gyrate atrophy in some way or another. Some of us have the disease ourselves, some of us have family members, some of us just have friends or have some familiarity with it. But many of us actually don't even fully understand what the disease is. And if you could just share some of the basics, particularly what do we know about the disease and what do we not know about the disease?
Sure, and just let me say, first of all, I'm going to defer this question to Dr. Uyhazi, who is the real clinician studying gyrate atrophy. And so she will be able to give you the best description. But I do just want to say one thing before we begin talking, and that is our research is really early stage. And we're in fact still planning many of the experiments. So we won't be able to give you a huge amount of detail in terms of specifics today. I view this as an introductory meeting. And as time progresses, we'll be able to fill you in as the studies progress. But I view this as a starter. But go ahead, Katherine.
Sure, no problem. So I'm Katherine Uyhazi. I'm an ophthalmologist at the University of Pennsylvania, specializing in inherited retinal diseases. And I'm also a scientist. So I have a PhD in stem cell biology, and I'm very interested in developing treatments for inherited retinal diseases like gyrate atrophy. And so my lab, some of whom are logged on today, as well as Jean, who I had the great fortune of training with, are currently working on several projects, including gyrate atrophy. And so it's really wonderful to be invited here today. So everyone knows, in this format about gyrate atrophy, but I'm happy to give you just a brief overview, a summary of this disease and what we know and what we don't know. As you probably know, so gyrate atrophy is an inherited retinal disease. It's autosomal recessive, meaning that you need one copy of the mutation from mom and one copy from dad. And when you have both copies together, you don't produce a functional protein called OAT, which I'll get into in a little bit more detail in a second. So this is a rare disease, as you know. It's hard to know exactly how many patients are affected because much of the time it goes undiagnosed. But rough estimates globally are that it may be around 1 in a million, 1 in 1.5 million or so individuals. For probably genetic founder reasons, it's much more common in Finland, actually. About a third of all of the known cases so far are of Finnish origin, and the frequency in that population is much higher, around 1 in 50,000. So the disease itself, it's very variable in terms of the age of onset. Most commonly, the first symptoms are in early childhood, but that can range from infancy all the way up to 30s, 40s. First things that patients notice are we can diagnose nearsightedness or myopia in childhood. Also night blindness, so difficulty seeing in dim conditions or nighttime. And then there's a progressive loss of peripheral vision in this disease. And so you may have loss of side vision that then gradually encroaches, and eventually later in the disease start having trouble with central vision. Patients with gyrate atrophy also can develop cataracts earlier than the typical cataract you would think of, oftentimes in someone's 20s or 30s. And like I mentioned, this is a very slowly progressive disease, but it does cause cell death in the retina and then eventually results in the loss of side vision, as I showed you. So what we know a lot about gyrate atrophy, and we know the genetic cause of the disease, which is mutations in a gene called OAT, which stands for ornithine aminotransferase. And basically how the system works is that part of the urea cycle is you eat food, you eat proteins, and one of which is arginine. And a natural part of the cycle is that arginine is broken down into some products, including one called ornithine. Typically, ornithine is then broken down by this enzyme that's mutated in gyrate atrophy called OAT. Ornithine is broken down into some other byproducts, glutamate and proline. And the problem happens is when you don't have a functional copy of the OAT enzyme, you can't break down ornithine. And so ornithine builds up to excessive levels, 10 or 20-fold higher than normal in patients with gyrate atrophy. And this is thought, for reasons we don't entirely understand, to be toxic to the retina. The excessive ornithine levels are actually everywhere in the body, but really the only place that it affects is the eye, and the retina in particular. And there are some case reports and some rare reports of the disease affecting some subclinical, meaning not symptomatic, but some incidental findings in skeletal muscle and in the central nervous system, and also in some hair. Many patients have fine hair. But overall, the main problem with gyrate atrophy of this deficiency of OAT is really a retinal problem causing the vision loss that I mentioned. And so the diagnosis is really a clinical one. The eye looks very characteristic, and I don't know if I can share any slides or not to show you a picture, but the eye has.
You should be able to share.
Let me see if I can. Let me see if I can pull up a picture just to show you. It says host disabled participant screen sharing, so that's okay. Basically, the eye has these very characteristic circular punched out lesions in the periphery. And they start off as little small circles, and over time and with years, they tend to coalesce and connect to each other and then form a large ring of atrophy or dying or lost cells with still an island centrally that looks like normal retina with preserved vision. Sometimes
I just enabled it in case you want to do that. Sorry about that.
Oh, no problem. Let's see if I can share this. Are you guys able to see that picture? Okay, great. So on the left-hand side here is is a picture of a normal retina. And if you're not familiar with looking at these, the central, I'm not sure if you can see my mouse, but there's a optic nerve right in the center, which is where all of the neurons in the retina connect and go back like the cables that connects to the brain that allows us to interpret visual images. All of the blood vessels emanate from that optic nerve. And then there's a kind of a dark circle in the center, which is the macula. And that's the region that we use for high acuity vision in terms of reading or seeing faces. And so if you look at the picture on the right, of course, this is from a patient, just a representative image of a patient with gyrate atrophy. And you can see these kind of classic appearing circular atrophic lesions, these punched out spots around the edges of the eye in the side vision. And those will come together and, like I said, coalesce, but the center part, the macula, tends to stay intact until very late stages in the disease. Just one more picture to show you, just to put an image to help everyone understand. This is that cycle I mentioned, and this has been well known for 20 or 30 years now. You eat the protein arginine, it gets made into this ornithine, which is shown centrally. And then the enzyme that is the problem in this disease, which is called OAT, circled here in red, is what usually breaks that ornithine down into other byproducts. Without OAT, ornithine builds up and you have retinal toxicity, retinal problems. So I won't, bore you with other details at that point, but let me stop there and open it up to additional questions if you have, and then we can kind of go into more details into what we're hoping to do in the future.
Thanks so much, Katherine. And I'll just point out on this diagram, I know some people are using B6, and so I just wanted to point out that that is used, I guess, in the metabolism of OAT. So some people are familiar with the fact that B6 works, I guess, in some of the mutations.
Yes. So
correct me if I got any of that wrong.
No, absolutely. B6, it does, like you said, works in a subset of patients, and we don't really understand clearly which ones. So typically there's a trial done of B6 supplementation and then ornithine testing after that to see if ornithine levels will decrease in response to taking vitamin B6. B6, it's a precursor to a protein that binds to OAT, so it's related in the same pathways. And there are some reports in the literature of certain mutations may benefit more than others with B6, but we really don't have a clear understanding of that. And so it's more or less a trial for patients to see if you're going to be a B6 responder or a non-responder. I should mention along those lines that current treatment for this disease, as you likely know, is really dietary. There is because of the pathway I showed you, the treatment is an arginine restricted diet in the hopes that if you can reduce the amount of arginine enough, you will make less ornithine, which seems to be the problem that's building up. And in order to do that, as you probably know, it's enormously difficult to maintain this strict diet. In order to limit the protein so much that you can get to the right arginine levels, you have to limit enough other proteins so much that you have to supplement with proline or others for their function. And so you also have to keep ornithine below a certain level, around 200, really consistently. If it goes above and below, and it doesn't really work as well as if it's consistently below. And so it's incredibly difficult to do that. And so it's another reason that we're trying to develop a treatment that would be much more doable, one-time treatment to replace the missing part rather than try to go before it and get it through just dietary changes.
Great. Thank you so much, Katherine. This is so helpful. Jean, I wanted to turn the next question to you. And I think many people on this call will have heard of Luxturna. But there may be a few people who have not. And, if you could just maybe tell us the story about what Luxturna is, how it came to be, and importantly, why folks in the gyrate atrophy community should be interested in that.
Absolutely. So I've been involved in the gene therapy field for more than 30 years. Starting at a time when we didn't know any of the genes which caused retinal disease. We had no way of delivering genes to cells, either in a dish or in an animal or into the eye. And we had no natural history. We didn't understand how the diseases progress. We didn't have surgical methods we could use to deliver genes to different cells, including the eye. All sorts of measures of how things could potentially improve, or even safety measures. And all of that had to be developed. And so in the 1990s, there was a lot of progress in developing, or at least understanding, the genetic basis of different forms of retinal degeneration or retinal disease, a large part due to the Human Genome Project, which was very active at that point in time. So we went from basically knowing about none of these diseases, including ornithine, including gyrate atrophy. That was the gyrate atrophy gene, the OAT gene, actually happened to be one of the first genes to be identified that causes retinal degeneration. That was about 1988, something like that. And there were, so there were a lot of steps to carry out to be able to do gene therapy. To do gene therapy, you need the genes, first of all. And you need to know what the genes are, and then you need to know how to deliver them. And so the second problem was delivery. DNA is highly charged, RNA is highly charged as well. And so you can't just apply it to a cell and hope it will get through the lipid bilayer. You have to play tricks on it. And there are various physical, chemical methods you can use by neutralizing the charge and so forth, but that's very inefficient and it's very transient if you can get the gene into the cell. So there was a huge amount of effort to be able to develop what are called vectors, and namely recombinant viral vectors that can piggyback genes into cells. They're essentially a Trojan horse, except these viruses are neutered, so they can't replicate once they get into the cell. So for example, if you're taking the common cold virus, you neuter it so that it can't cause the cold, and stuff it with the gene of interest plus an on-off switch that's called a promoter or regulatory switch. And that way you can actually get the gene safely and efficiently into the cell. A lot of the reagents that were used initially as vectors, the recombinant viruses, were cleared by the immune system, but there happens to be one virus called adeno-associated virus, which even in its wild type form, the unneutered form, has never been shown to cause disease in humans or animals. And that is very efficient at delivering genes to cells without engendering a huge immune response. And so in about 1995, we were one of the first groups to test adeno-associated virus for delivering genes to the retina. This happened to be a mouse, and we were using what's called a reporter gene. It's a gene which turns the cells a different color, so you can actually see whether the gene is working. We showed that this worked well in a mouse, and voila, that set off a whole new path of realizing, yes, we can deliver these genes to the cells of the retina and they stay there, they stay functioning. So if we then extended that study to a monkey, for example, we could inject the gene into the cells of the macula and then look 2 years later, and those cells were just as green as they were 2 years earlier, without any disease and without impairing the normal function of the retina. So we knew we had something. And in the meantime, with the progress of the Human Genome Project and the ability to identify animals that had the disease, we were really lucky to find that there is a dog, a Swedish Briard dog, which is a big, hairy dog. It's about the size of a horse, and it's a really sweet animal. The hair comes over its eyes, and at first we thought, wow, this dog can't see because its hair is covering its eyes. Not true. The dog can't see because it was missing a gene called RPE65, stands for retinal pigment epithelium 65 kilodalton protein. And the gene had just been identified in humans to cause congenital blindness. And the dog, people took the next step, realizing, well, maybe the same gene causes the same disease in dogs. And lo and behold, it did. We happened to have 3 of these affected dogs in the colony at University of Pennsylvania. And we had cloned the gene and put it into this recombinant virus vector, and we tried it out on these 3 little puppies. These little puppies would walk around. They were scared to walk around because anytime they'd walk, they'd bump into something because they couldn't see. So they'd crunch in the corner. Behaviorally, they just looked miserable. They had to be shown where their water bowl was because they couldn't see it, and they wouldn't play with each other, very unlike other sighted puppies. We injected 1 eye of each of these puppies, and I went home, and then a couple days later, I got a call from the animal technician saying, "Gene, these dogs can see. They're following me as I walk around the facility, changing cages, and I bring them out of the run, and they're playing with each other, and they're catching things, and they're walking around without bumping into objects." I couldn't believe it. I hightailed it into the facility and checked myself, and then, of course, we did a lot of objective physiological studies to verify, yes, indeed, these eyes were seeing now. And that was the whole beginning. It was a eureka moment. It was really a stunning moment. And of course, the first question that arose after that was, wow, wouldn't it be great to be able to harness this technology to make blind children see? And so that allowed us to embark on a whole series of studies, which brought us to clinical trials and then ultimately to drug approval. The entire process, I have to tell you this, everybody, don't gasp when you hear how long this took. From the time we injected those puppies to the time the drug was approved, Luxturna was approved, it took 17 years. It could have been a lot faster, but it just so happened that the first studies that we did took place around the time when there was a really bad incident in the gene therapy world, and everybody thought, oh, this is never going to work, and all the funding dropped out of it, and companies that had been established to support gene therapy all folded. And so we had to kind of slog through about 4 years when people just weren't interested in gene therapy and just didn't think it was going to work. We knew it would, and so we kept going. But I'm really excited about the potential for gene therapy for gyrate atrophy. And I'll stop here to take any questions, but I can go on and tell you about the next steps for clinical trials if you want.
Thanks so much, Jean. I have just a couple more questions, but audience members, if you have questions, feel free to put them in the chat. And like I said, I'll be sure to leave time so people can ask their questions as well. This is, I guess, a question to both of you, sort of building on, the success of Luxturna. Can you talk a little bit about how you're approaching the research for gyrate atrophy, and maybe share a little bit about just sort of where you are in the process. And I know it's early days, but whatever you can share about where you are now.
Sure. Yeah, I can start off. So we're really excited to try to tackle this problem. Now that Luxturna is approved, we're as a field really, really hopeful that this has kind of paved the way for the development of other similar therapies, gene therapies for eye diseases. We know that it works well for RPE65, and when Luxturna is approved, we know a lot about delivery. We know a lot about the viruses now. We know a lot about that it's safe, and what to really look for and test. And so we're really hopeful that that will expedite things and that we can use that technology to develop similar treatments for other diseases, including gyrate atrophy. So there, one important thing is that we know the gene and we know a lot about the disease already, which I mentioned. And so that's kind of the first hurdle, is that many eye diseases we would love to develop treatments for, we simply just don't know the genes. A lot of diseases, so macular degeneration is a huge problem, but we don't know the genes. And even if we could find some, it's most likely caused by multiple genes and isn't really a good target for gene therapy. Gyrate atrophy, on the other hand, we know the gene, we know the protein, we know what it does. And so it provides a nice window and opportunity for us to try to apply the same exact technology of treating RPE65 mutations, but with replacing the OAT gene instead. And so our hope is that we would use very similar techniques but with putting back a normal copy of the protein that has a typo in the DNA. To do that, so we, and I should mention too that one of the nice things about the OAT gene compared to other genes that we even know is it's technically possible. There's some technical problems of developing these gene therapies, and OAT doesn't have some of the size problems that other genes may have. So the real goal for us is to first really characterize the human disease well. We want to make sure we understand all of the details of how the human disease looks, how the retinas behave and how they function, so that we can understand and have a really good baseline of what we're trying to improve. It's hard to tell if a drug or gene therapy is making a difference and an improvement if you don't really know where the disease naturally goes. And so natural history studies of everything tends to be step one. Step two then is testing a potential treatment in animal models. And so whether it's mouse or dog, we typically use mice in the lab, and we decide, we try to develop what we think is our best guess at what's going to work the best to develop a treatment to replace this gene, and we test it in animals to make sure it's safe, number one, before even thinking about clinical trials, and also hopefully to see if it's effective and to make sure we understand what it's doing, it's safe, and how well it works, to give us clues about how to progress to the next steps. If all goes well in animal models, like in the mouse, then we would typically switch to dose testing and safety testing in a more relevant animal model to humans. So as you know, the mouse eye is a lot different than the human eye. One of the biggest differences is size, of course. Mouse eye is much smaller. Mouse eyes also don't have a macula though, like a human eye. It's that central region that I mentioned and showed you in the picture that's responsible for high resolution vision. And so especially in this disease where the side vision is more effective than the central, it's hard to mimic everything in that disease in a mouse that doesn't have a macula, doesn't have that same anatomy as a human eye. And so typically we go to a monkey to test the safety of a potential treatment therapy. And the monkey doesn't have the disease, so it would not have the same problems as a patient with gyrate atrophy or a mouse with the disease, but it's used to make sure that what we would inject is safe and doesn't cause any problems. And then if all of that looks good, then we would progress to the next step, which is proposing a clinical trial to test safety and efficacy of a potential treatment. And I'll turn over things to Jean, who knows much more than I do about the timeline and the details of clinical trials, and maybe she can give you an outline of what to expect.
Sure. Of course, the number one question, and I see one in the chat, is about the timeline. How long would it take to figure out a cure? And that's a very, very difficult question to answer because there are all sorts of unknowns. There are many, many variables that we want to test. And of course, we want to make sure that this is safe, that there's absolutely no question about safety. And in fact, I can probably speak for Katherine as well, that our gold standard of safety and efficacy would be higher than any rating that the FDA had. It would be, if our children had this disease, would we allow them to participate in a human clinical trial? And that really was my guiding light when we were involved with the whole Luxturna studies. But there are a whole number of variables that we will be sorting out in the preclinical studies. The first to show that we can actually reverse some of the deficits in models of gyrate atrophy. Then looking at the appropriate dose, making sure that the effect is stable, and then looking at safety from every which angle. And then doing a lot of boring things that you have to do in clinical trials, like make sure that the reagent isn't sticking to the inert surfaces inside the device, inside the syringe, for example. We have to make sure that what we put in, the dose we put in the syringe, actually comes out of the syringe so that we're getting accurate readings. We have to figure out ways of showing in a dish that our reagent is working and that it doesn't lose its punch over time with storage, for example. So we would generate the clinical reagent and put it in the freezer and then take it out take out aliquots at various time intervals and test to make sure that it still has what's called potency. Before we can do that, we have to develop a potency assay to standardize this outcome measure. And this is no trivial problem, is to go through the enormous amount of work it takes to get regulatory approval to move forward with a human clinical trial. And that basically involves two different bodies. One, it involves the FDA. And in the process, the conversations with the FDA can begin early on to get feedback about your plan. That's always a good idea to get feedback early on, particularly if they want you to change something, because you don't want to go for a couple of years and then be told, no, you can't plan on this kind of approach. So we have conversations with the FDA. Those are formally called pre-IND meetings. And during that sort of conversation, we would ask the FDA if it's okay with them if we proceed with this preclinical toxicity plan. For example, if we have our reagent, we show that it works in a dish, can we then test the safety of that reagent in monkeys? And how long do we have to wait? What assays do we have to use to make sure it's safe? Typically, those assays involve what are called electroretinograms. They measure the electrical activity of the retina after it's exposed to light. They involve regular eye exams, measurements of pressure of the eye, photographs of the retina, and ultimately histology to make sure that there's no immunologic response and to make sure that your gene of interest is still working in that retina. And you typically do it at a higher dose than you plan on using in humans because you want to see the worst possible outcome. You want to make sure that this is going to have robust safety profile. And once you've got all of those results, and those all have to be done at a certified research organization, they're what are called good laboratory practices study, GLP studies, where there are people watching every allotment of pipette, recording the serial numbers of this, that, and the other thing, making sure nobody's interfering with the results and that there's a chain of custody with the reagents and so forth. And the analyses aren't done by us. They are done by people who are neutral. They don't know necessarily what the reagent is or what the dose is and so forth. And they generate a report. And presumably that will look good in terms of the safety profile. At that point, we would go back to the FDA and submit what's called an IND, which is investigators' new device protocol or procedure protocol. And the FDA then has 3 months to review it. If you don't hear from them, that's a good thing. That's a green light. And that means that you can proceed with your clinical trial. But in the meantime, during that 3 months while you are waiting on pins and needles to hear from the FDA, you go to your institutional review board, which is abbreviated IRB. There are tons of abbreviations in this whole process. There should be a whole dictionary of these abbreviations. And you apply basically for an IRB. It's an institutional review board approved protocol to enroll human subjects. Beforehand, you've decided what age of people that you want to enroll, and that would dictate whether you're going to do it at a children's hospital versus an adult hospital. You write out specifically what you plan to do, what doses you're going to give, and typically people start at a low dose, then they go to a mid dose, and then a third dose in different cohorts of patients. You have to write inclusion, exclusion criteria, who would be eligible for this and who wouldn't. So for example, the people who would be eligible would have to have bona fide OAT mutations to be enrolled in this study. And people who might be excluded are those who don't even have any retinal cells left because the disease has progressed so far. That's the kind of inclusion-exclusion criteria we would set out. And that institutional review board is composed of something like 24 people from all walks of life. They're physicians, they're scientists, they're administrators, there are laymen, often there are patient advocates, for example, somebody from a family with gyrate atrophy, chaplains. And these people have very different concerns. Our IRB board told us that we would have to make sure that all of the subjects in our Luxturna studies use contraception for a year after having their gene therapy. And we, as a result of that, our IRB consent and assent protocol, each one of the children who was enrolled had to agree that they would not father a child during that first year, which brought some amusing conversations with 8-year-old boys. So there's some funny things that happen along the way. But hopefully the institution approves the study at that. And by the way, there are additional things besides the informed consent and so forth. At our institution for Luxturna, I had to go through 13 different committees before we got institutional approval. There was the device committee, the conflict of interest committee, the pharmacy committee, the this, that, and the other committee. There was even a committee overseeing the committees. I think it's eased off a little bit since then, because when we first started, gene therapy was really an unknown, and it's been de-risked a great deal, not only at our institution, but around the world. So hopefully it won't be as tedious, but hopefully the timeline then coalesces so that the IND gets the green light, the institution gives the green light, and then the next step is that patients can be recruited. They cannot be recruited to the trial until then, but once that has occurred, then there can be, for example, a posting on Facebook or little handouts that would go out, or the Conquering Gyrate Atrophy group would get a message saying, okay, we're ready to enroll, spread the word. We can't enroll, we can't accept patients until that point in time.
Thank you so much. It's so daunting when you think about all of the various steps from start to finish. It's really daunting, which just makes Luxturna that much more impressive as an accomplishment. We are getting a number of questions, so I want to make sure we go to them. And maybe Graciela, if you want to ask your question, and then I'll go to some of the questions in the chat. Yeah, go ahead.
Can I talk? Okay.
Yeah.
Okay. Hi, Jim, Bennett, and Katherine. Well, I'm Graciela. I have gyrate atrophy. I just get my degree as a medical doctor. And well, I'm the founder of a foundation in Mexico of gyrate atrophy. I have patients of gyrate atrophy from Latin America and Mexico. And well, I'm so glad to know you. And well, I just have it's not a question, but it's a finding that we found at Instituto Oftalmológico Conde Valenciana in Mexico. We have collected 7 patients of gyrate atrophy, and we are preparing paper. So I just want to read this finding that we found. So maybe it's going to be helpful for you. And well, if it's too obvious, just let me know. But I think it's a good finding for gyrate atrophy and for all these. And well, I'm so happy and excited for this. Okay, in Mexico, we have found that most of our patients with gyrate atrophy have an outer retinal tubule or outer retinal tubulation in the OCT. Those who have ornithine levels, those whose ornithine levels are high. We have found these outer retinal tubules, and one patient whose ornithine levels was normal these findings were absent. Well, the thing is that we are so impressed about these outer retinal tubules, and I don't know if this finding can help with the gene therapy when you apply it and all these things. I don't know if you have heard about it.
Yeah, thank you. It's really wonderful to meet you. Congratulations on your degree, and it's really great to get to know you. So the retinal tubulations, it's not a specific finding. We often see retinal tubulations in other diseases of the retina as well, but it could be useful. And like we said, this is very early, and we're not eliminating anything. So the tubulations occur, and this is a kind of a fancy word for those of you who aren't familiar with this field, is that the retinal cells, the photoreceptors in the back of the eye that are actually the cells there that see light and transmit that signal into vision that you're interpreted by your brain. Sometimes the cells don't know how to line up perfectly. And when that happens, whether it's due to a mutation in OAT for this disease or in other inherited retinal diseases, sometimes they make these little circles because they just don't know where to go, and they don't have the right signals to line up like a fence perfectly as they normally do. And so that's not specific to gyrate atrophy, but it's a general sign that the retina is not healthy. And it's a great suggestion, and we'll certainly look at that on the imaging, and hopefully can correct those types of defects.
And just to add, we look forward to reading your paper.
Yes.
There's another gene therapy clinical trial that we ran for a disease called choroideremia, and retinal tubulations are also present in that condition. As Katherine said, it's common in many different diseases. So it wasn't an exclusion criteria for participating in that trial.
Great, thanks. So I'll go to a couple of questions that are in the chat. So one question is, given that gyrate atrophy is a metabolic disorder that begins in the urea cycle, are there studies or trials in animal models with gene therapy to examine or study the liver rather than focusing primarily on the retina and eye functioning? I don't know if either of you can comment on that.
I'll start. I'm not aware of any studies targeting the liver. They may be happening. One thing about science is we usually don't know what's going on until it's published. I do know that people long ago, one of the first planned clinical trials for choroideremia was to transplant skin cells to the thigh of a person. In other words, to do something far away from the eye, and to be able to ameliorate the disease. The liver sounds like a good target too.
Yeah, we really don't know what's going to work best, which is why there's no treatment yet, but we're trying a lot of different approaches. And the eye is kind of a special organ in many ways because it's somewhat protected from the immune system. And so that's one of the reasons why Luxturna has been so successful, but we really don't know. And it's a great question to kind of keep in the back of your mind.
Yeah, and I'll just add that from the perspective of conquering gyrate atrophy, we are trying to kind of raise funds and stimulate research along a number of different areas. And Jean and Katherine's research is certainly one really compelling area. If you know of researchers who are interested in another research direction, then we obviously would like to hear about it, because we don't know which, that's why it's called research. We don't know what's going to eventually be successful. So all right, there's another question, a good question. So would a cure for gyrate atrophy, similar to Luxturna, correct retinal cells that are already damaged versus just prevent progression? And I don't know with Luxturna how it works, but if you could speak to that.
Sure. Well, for gene therapy to work, you need a cell that's alive. It can be sickly, and the gene therapy could potentially enhance its function. If the cell is moribund, i.e., there's nothing you can do to revive it, it's probably not going to help it too much. But so I would hedge and say it's probably going to depend upon the stage that that particular cell is going through, where it is. Katherine, do you want to add?
Yeah, I mean, I agree. I think the real answer is we don't know how it will work entirely until we try it. Even if we do it in a mouse, it doesn't necessarily line up to what we would find in a human until we try it. But in general, you know, the gene that goes into the cell directly can improve function in that cell, but it can't bring back cells that have already died. And so stem cell research is another really exciting, really promising field to be able to try to get cells, not just to slow down the cell death and stop the disease, but reverse it and try to bring vision back. And so our group is also working on stem cell research and trying to get cells to turn into the eye cells to be able to replace them. But that field in general is much farther behind the gene therapy field, and we're working very hard, but are, you know, about a decade or so behind gene therapy, I would say.
Could I follow up with a quick follow-up question to that?
Sure, go ahead.
So I thought that might be the answer to one. That was my question. So I'm not sure if you've ever read about, there's two scientists, they kind of work in different areas, not on eye diseases or like eyesight, David Sinclair and Andrew Huberman. And so with the David Sinclair one, he focuses more on like aging stuff. But anyways, he had a clinical trial where they had the unintended effects where they were testing on mice, where they were in a very similar way of Luxturna and stuff, where that viral mechanism was inserted. And what they found in these mice is that these old mice who had really bad eyes, their retinal cells started growing again and whatnot. And it was like they were amazed by something along those lines could happen. So it's really interesting. 20 some odd years ago when I was first diagnosed with GA, and I thought to myself, and I'm not a scientist, and I thought, you know what, this, the diet thing for me, I said was ridiculous, because I'm like, great, so I can extend my vision and die of a heart attack with all the high fat diet. Anyways, and I'm like, well, if the problem is, so this problem should be two parts. One is that you have the disease that damages the retinal cells. And if something like a GA equivalent will stop that, then part two is, hey, is there any way I can fix my retinal cells? So, I mean, and in my case too, I'm pretty lucky. I'm 45. My vision is still very useful. I still drive. Nighttime's awful. It's pretty much like night sucks. But yeah, so I'm that's why I'm curious about that. I don't know if those two names and stuff has if you've ever come across that in any sort of scientific research or anything.
Jane or Katherine? Yeah.
I'm aware of David Sinclair's work, and I find it fascinating too. And it's kind of like the fountain of youth. You're right. It's an area to follow and see how it proceeds.
Yeah, I think sometimes nature just amazes us. You know, there have been patients treated with Luxturna who had very little to no functional retina, but then actually regained quite a bit of function after treatment. And we may not have predicted that, but it happened. And we kind of have to work backwards and figure out how it works and then try to get that to happen in other diseases. But despite our best understanding and best predictions, sometimes nature just tells us the real answer. So I think it's a really fascinating field, and I hope that you know, moves forward, but we just don't know yet.
Thanks so much for your time. Appreciate it.
So another question, and I'm sure this question is on a lot of people's minds, and you started to answer it, Jane, but I think there's a lot of folks in our community who are just eager to you know, support the research and get the research done as fast as possible. And there's a question in here of, would it work for your studies if we sent you our genetic test results that to help with your database? And so I know one part of the answer was we need to wait till there's clinical trials and recruitment, but are there things that GA families could be doing now in terms of, you know, providing data or anything for your own research?
Absolutely, there are things that you can do, and we're the whole patient community is indispensable. This is where it all starts, and where we want it to end and improve the condition. We are not allowed to recruit information like that now, as I said. However, there's nothing to stop you from sending it to us. And in particular, Katherine, who is the clinician, it could be very useful. She may have studies down the line to evaluate different outcome measures that she's thinking about for a clinical trial. It's really helpful to try them out on somebody with the condition before actually setting in stone what you're going to do in a clinical trial. Because once you start that, you can't change the protocol. We in the whole Luxturna experience, patients helped us every step of the way. We had to justify including children in our studies in front of what was then called the Recombinant DNA Advisory Committee, which is run by the NIH. And up until that point, no one had ever enrolled a child in a gene therapy study with a non-fatal disease. And we had families come to that meeting and describe how difficult it was for them to get through every day with their child, and why they would take the risk of allowing their child to enroll in a gene therapy study. And after that testimony, first of all, there wasn't a dry eye in the house. But second of all, the committee unanimously approved enrollment of children in gene therapy studies, and that had a cascading effect on other studies that now enroll children. And we'll need you to talk to the FDA. You know, like hopefully we get to the point, or Katherine gets to the point of going to the FDA with the huge package of data from the clinical trials that looks great, that looks safe, it looks like it's benefiting people. And at that point, the FDA sets up a scientific advisory committee that evaluates this data really carefully. And patients with the disease come and give testimony about what they think. That's another area where we could really use your help. And further, there may be studies involving cells, be able to study the biology of cells or the effects of different mutations of the cells, and to be able to predict how they'd respond to treatment. It may be that that investigators would be able to benefit if you were willing to provide a blood sample or a little skin biopsy. So there are lots of ways.
Yeah, I would be happy to provide my contact info to maybe Conquering Gyrate Atrophy. Maybe is the most central location if you were interested in sending info. More importantly is that you at least have your genetic mutation, I would add too. So if you haven't already had testing to know the specific mutation in OAT, that's something worth doing early, even before any trials are even on the radar, because that would be a requirement to know what your mutation is before you'd be eligible to be enrolled in most of the studies. But yeah, absolutely. You know, you guys as a community is really critical to making all of this advance. And so if we have a group and have a database of everyone involved and interested in participating in research studies, then we can easily reach out when we have a need to make things move forward.
Great, thank you so much. We have time for just maybe one or two more questions. If you want to put it in the chat, feel free. In the meantime, I have a question for you, which is, you know, even in this group, I don't know how many countries are represented on this call, but I think it's many, definitely more than 5 or 6, maybe as many as 10. And could you speak a little bit about so if the FDA approves it, and that's only in the United States, how does that then work for other countries? How do non-US nationals participate in clinical trials? Could you just speak to sort of the international dimension of how this might work?
Sure. Yeah, there's no exclusion of people outside the United States being able to participate in a clinical trial in the US. In fact, for Luxturna, our first 4 patients were Italians, and their way was paid to come back and forth for the multiple visits that they came and for the surgery and so forth. And I would say probably half of the patients, well, definitely half of the patients in phase 1 were foreign. So that's not a problem. As far as getting you have to go through this similar or analogous regulatory steps with the Europe, with those countries. For example, when we enrolled the patients in Italy, before they could be enrolled, we had to go through what's called the ethics board, which is the equivalent of the IRB in our institutions, and basically they used all of the answers that we had supplied our IRB. So it wasn't too rigorous. For getting approval in another country, for the European Union, the equivalent of the FDA is called the European Medicines Agency, the EMA. And they have a very similar process as the FDA. In fact, we went through both processes simultaneously, both the FDA and the EMA, so that Luxturna was approved in both locations. And they have slightly different focuses. For example, the EMA really wanted to make sure that we would treat infants as soon as they were diagnosed, which is something we hadn't planned on doing. The youngest child that we enrolled in a clinical trial was 5 years old. And the net result, however, was when the drug was approved, it was approved for age 1 and over, and that's thanks to the EMA. I don't think a 1-year-old has been treated with Luxturna yet, but that may happen soon. And similar agencies exist for other countries. One of the most rigorous is Japan. They have a very rigorous approval process, but the others are pretty similar to FDA.
Thanks so much. Okay, one last question, and I think Katherine, maybe this is for you. Where are you now with the studies, and what are you going to do next? What can you share with us?
Yeah, sure. So right now we're taking our best guess at what we think is going to be the most effective treatment for this disease and testing it in a mouse model of gyrate atrophy. Gyrate atrophy is a very slowly progressive disease, as you know, in people, and that's also true in the mouse. So we can't just take a mouse and wait a couple of weeks and see if it works. It takes up to a year really to see retinal problems, just like in a human. And so it's slow going, but right now we're developing what we think is our best guess at working, and then testing it in these mice. And it's going to just be waiting to see what we can find. And of course, we're excited to share any exciting data as we have it.
Thanks so much. So I'm seeing a number of questions about sharing data. So I think what we can do there is I can maybe connect back with you, Katherine, and figure out what is the best way to do that. This
Sure.
is, sensitive data. So what I will do is I'll connect with you, Katherine, figure out what would make sense. And then I will send a note out to everyone who's been on this call and just share, if you would like to share your data, what the best way to do that is.
Sounds great.
So, yeah. So we are almost at time. Katherine and Jean, I just want to thank you so much for participating in this discussion. It means the world, I know, to all of us in the community. Just to know that that you're working on gyrate atrophy. I mean, I remember a few years ago, no one was working on gyrate atrophy, and you guys have hit the ground running and are going at warp speed, and it's so exciting to see. And I just thank you so much for sharing that with us today. And I want to thank everyone for coming. This is beyond our wildest dreams in terms of the number of people who've participated today, given how small the patient population is. So thank you so much for coming. And this has been recorded, and it will be up on our website in a little bit. So thank you all so much.
thank you all so much. It was a pleasure talking to you, and thank you for joining forces with us.
Likewise,
Thank
thank you
you.
so much, and hopefully we'll see you soon.
Yes.
Thank you very much.
Bye-bye.
Thank you.
Bye.